The human reproductive process is a complex system with many processes including but not limited to spermatogenesis, oogenesis, ovulation, fertilization, and gestation. Our focus is fertilization, more specifically the process by which the sperm enters the egg. Generally, this process occurs in vivo or in the body. However, in certain cases, this will not occur naturally. In these cases, people may choose to use in vitro fertilization. In vitro fertilization, or IVF, is an example of an assisted reproductive technique, ART, in which an egg is inseminated in a laboratory and then transferred to the mother's womb. Let's look at the case study of a 43-year-old man named Robert with Cartagener syndrome. Cartagener syndrome falls into a group of disorders called ciliary dyskinesias. These disorders are characterized by defects in the movement of cilia lining the respiratory tract and the flagella of sperm cells. To diagnose him, the doctors analyzed Robert's semen 3 times. This analysis found that Robert had decreased viable sperm with no motility. Because of this, Robert had no children despite being married for 15 years. Nevertheless, Robert and his wife want to have children. So what should they do? After the doctor diagnosed Robert, she suggested he and his wife use intracytoplasmic sperm injection, or ICSI, a type of in vitro fertilization therapy.
Intracytoplasmic sperm injection, or ICSI, is a reproductive technology used to treat sperm-related infertility problems, where in vitro fertilization encourages the most fit sperm to fertilize the egg. ICSI enhances this phase by injecting a single selected sperm into a mature egg. The fertilized egg is then placed in a woman's uterus or fallopian tube. In the conventional process of fertilization, a fertilizing spermatozoon must pass layers of cells and then make contact with the zona pellucida, the glycoproteinic envelope that surrounds and protects the oocyte. When the sperm meets the zona pellucida, there must be recognition of specific receptors on both the plasma membrane of the sperm and the zona itself. In order to penetrate the zona, a spermatozoon must go through a process known as the acrosomal reaction, which releases enzymes that enable the digestion of a path through the zona. After penetration of the outer layers, the spermatozoon must then fuse with the plasma membrane of the oocyte and enter the oocyte, which eventually leads to the formation of the embryo. Because ICSI involves the injection of a single spermatozoon directly into the oocyte, this procedure bypasses all the preliminary steps of fertilization. Matured eggs are obtained from the woman by hormonal ovarian hyperstimulation. Then, the best sperm are selected on the basis of morphology and mobility.
Back to Robert. In males with Cartagener syndrome, simple IVF is a common treatment. However, this is only in cases in which sperm retains some motility. If you recall, Robert's sperm is viable, but completely immobile. Putting the sperm in the same dish as the egg is useless if the sperm can't make it into the egg. Therefore, ICSI, injecting the sperm directly into the egg, would be a better treatment.
Is this enhancement of in vitro fertilization promising? A paper by the Canadian Medical Association Journal analyzed the promises and challenges of ICSI and found that there is conflicting evidence about the safety of this procedure across different studies. A study in West Australia found that both ICSI and IVF were associated with increased major birth defects at 1 year compared with natural conception. In contrast, a large-scale study in Sweden found that the incidence of malformations in children born after ICSI was not significantly higher than in normal controls. In a case-controlled study in UK, Sutcliffe found no mean difference in neurodevelopmental scores between children conceived by ICSI and those conceived naturally. So, what are the next steps? In the future, large-scale studies that examine demographic, clinical, and laboratory data are necessary to find the relationship between ICSI and birth defects. Examining harmful fetal and infant outcomes and determining the association between ICSI and rare clinical conditions and controlling for confounding factors will help us provide the information necessary for infertile couples to make the best choice for them.
But what if Robert and his wife lived in an underdeveloped country? What if he was financially disadvantaged? Recent research from the journal Reproductive Biomedicine has an interesting find. A major setback of common IVF strategies such as ICSI is that each cycle of IVF costs on average $12,000. Usually multiple cycles are required for a pregnancy to occur, further driving up the price. This journal discussed a new device created by Jonathan von Blurkum that uses glass tubes, citric acid, and sodium bicarbonate that brings the price down per cycle to $250. This system works through an incubator system consisting of 2 sterilized glass tubes and a block of aluminum that heats the tubes to 37 degrees Celsius. In one of the tubes, citric acid and sodium bicarbonate mix with water to make CO2. These chemicals were chosen because of how cheap and common they are, and because they can mix to make the same stable pH in atmospheric conditions that medical-grade gases and microprocessor-controlled tissue culture incubators can. After the 2 chemicals mix, the gas is transferred to the second glass tube holding a culture medium. Oocytes and sperm are injected via syringe into the second tube, where they combine to form an embryo which is then implanted into the mother's womb. In the study, pregnancies resulted in 8 of the 23 patients. To date, 17 healthy babies have been born by using this system. Though the practice is relatively new, Robert can find comfort in the fact that plenty of time and effort is being invested in the facilitation of in vitro fertilization.
So why is this type of research so important? As technology advances, more and more people are able to reproduce regardless of what problems they might have previously encountered. Limitations that once hindered the biological fitness of individuals are being altered by the power of technology and cutting-edge research. As more people can reproduce over time, genetic variation in the population can increase. Undoubtedly, in vitro fertilization has benefited many families and revolutionized the field of science.